Welcome back, folks. This is chapter 13, part A, Glycolysis and the Krebs cycle. The Krebs cycle comes under many different names, and you'll see that as the TCA cycle or the Citric Acid Cycle. All mean the same thing. The learning objectives for part A are quite diverse. It's a rather long section, and it has many parts. But most importantly, students need to remember that the chapter is all about how cells extract energy from food using cellular processes. The food that enters the cells, in most cases, the preferred source of energy is glucose, and that process is called glycolysis, the subject of the first part of this video. And in the presence of oxygen, the food particles, as they are being manipulated, are then sent to the mitochondria, where they are subject to the TCA cycle, or the citric acid cycle, or otherwise known as the Krebs cycle. Once that process has concluded, the energy that's been liberated through both the glycolysis and the citric acid cycle is then harvested using another cycle, which is the subject of chapter 14. But for now, we need to learn a couple of things. The first thing is anaerobic respiration versus aerobic respiration. And this will be cellular respiration, not respiration within the lungs. So as the word suggests here, aerobic. Aerobic means in the presence of oxygen. So an means without. So anaerobic respiration is what happens to cells when they don't have oxygen, whereas the alternative is what happens inside cells when they do have oxygen available. So students should be familiar with that. Talking about being familiar, it's very important that students review chapter 3 from this textbook. Chapter 3 has at least 8 sections which we must be really familiar with. And as it says in my PowerPoint, the next 14 slides are taken as a synopsis of chapter 3. So students should be extremely familiar with these slides, otherwise they may find it difficult to relate to some of the concepts that are being talked about in this video and indeed in this chapter. The ultimate accumulation of your knowledge should allow you to reach this point, which is the first figure in chapter 13. If we focus our attention on panel B first, where the sugar molecule, in the presence of oxygen, can release its energy. However, if all that energy was released in one single step, one burst, that will be akin to an explosion taking place in the cytosol of cells. And that release of energy would be wasted, and indeed, if it happens often, as it does in the real case, the cell will increase in temperature and eventually burn up and fry. So that's not a good thing. So what nature has done basically is to release the energy in small steps, and each step is responsible for manipulating the sugar molecule in a way that will allow that energy to be released. Now, this diagram is not actually 100% correct in that energy is released in a downward spiral, each step releasing energy into the environment. In fact, energy actually has to be invested in the molecule first before that energy is released. But regardless, we get the idea that cells are very good at having multiple steps in a pathway, and each step in the pathway controls the release of energy, therefore making more of that energy available to the cell and not as heat. Many aspects of this chapter are relevant to all living cells. However, the primary focus appears to be on human cells, and indeed that's where the beginning of the process begins, with laying out that the process of extracting energy from food can be divided into 3 distinct stages. So stage 1 is digestion and absorption. So this takes place in the human gut. So using our teeth and our mouths and our stomachs and our digestive system, we digest food into smaller and smaller chunks until those subunits are small enough to be absorbed across into the blood supply. Once in the blood supply, they are then delivered to the cells. Once the molecules are uptaken by cells, they are present in the cytosol. So stage 2 is glycolysis, the breakdown of the food molecules into smaller and smaller entities with the breakage of the covalent bonds holding the atoms together. And that process will continue until the release of energy takes place in the form of the citric acid cycle. And that's stage 3. So stage 3 occurs in the matrix of the mitochondria, because that's where the enzymes are that can actually handle acetyl-CoA, the first chemical in the citric acid cycle, and then use that substance to break down further to release carbon dioxide and water. So the whole process takes place both in the cytosol, outside the cells, and inside mitochondria. Now, chapter 14 will allow us to then understand how oxidative phosphorylation takes place in the inner mitochondrial membrane. But again, that's going to be chapter 14. So chapter 13 stops at the production of water and carbon dioxide from the citric acid cycle. One of the things to keep in mind is how many ATP molecules are being made during the different stages of these steps. So paying attention to the words is very important. So in this case, it's asking not about the entire cycle, but just the second step, which is glycolysis. And in that case, the answer that you'll find is that for each molecule of glucose, 2 ATP molecules are generated net. In this particular figure, we're looking at how glucose, a 6-carbon sugar, is manipulated through a sequence of 10 steps, resulting in the production of 2 3-carbon pyruvate molecules, otherwise known as pyruvic acid. So because the bonds between these 6 carbons have been broken, energy must have been released. And that energy has been captured in the form of NADH, an electron carrier, and also another carrier molecule called ATP, which we're familiar with. Now, don't let this figure confuse you. What it's saying is that initially the cell has to increase the energy content of the glucose molecule to make it unstable. But once it's unstable, then the glucose molecule is split into 2 halves, and then from each half, energy is extracted. So overall, the production of 2 ATPs and 2 NADH molecules takes place. Now, nowhere along this pathway is oxygen required. Oxygen is required, as we'll find out later in the TCA cycle, or the citric acid cycle. So glycolysis occurs in all cells, more or less, and it takes place in the cytosol. So if we were to summarize what we just said on the previous slide, then these 3 boxes here would have that summary. So for steps 1 to 3, the first 3 steps of glycolysis out of the 10, energy is invested in the molecule to make it unstable. From steps 4 to steps 5, the molecule is broken into 2 3-carbon sugars. And then finally, from step 6 to step 10, energy is recovered from this molecule and passed on to energy carriers. Steps 6 and 7 receive the most attention because they confuse the students sometimes. So during step 6, an enzyme called glyceraldehyde 3-phosphate dehydrogenase, and if you break this down, you should be able to figure out what the enzyme is doing. But basically, the enzyme is responsible for charging NAD plus an electron carrier with some passengers. These passengers are 2 electrons and 1 proton. So 2 electrons and 1 proton. So the question is, why do we have the number 2 in front of these electron carriers? Well, if you recall, one molecule of glucose breaks up into 2 3-carbon molecules as it progresses down the glycolytic pathway. So we have 2 molecules of glyceraldehyde 3-phosphate from each glucose molecule. Therefore, everything is multiplied by 2 to account for those 2 molecules. Students are encouraged to learn the name of each enzyme at each step, the substrate going into that step, and the product of that step, which then becomes the substrate for the next step. But more importantly, students should relate to the logical manipulations of the molecules that are taking place. So here's glucose, and glucose, like we said earlier, is a 6-carbon molecule. Sometimes the 6 carbons can be depicted as a circle, other times as a linear molecule. And inside a cell, it resonates between those 2 structures. Regardless, glucose is a very liable product. That means it will diffuse through the GLUT transporters, the facilitated transporter proteins for glucose, down its concentration gradient. So if the level of glucose is high inside the cell and slightly lower outside, then the cell will lose its glucose through the gradient. So one way that nature prevents the glucose molecule from leaving is to change its identity. Therefore, it's no longer available to its concentration gradient. And one way to do that is to add something to the glucose molecule itself. And that's done by hexokinase. It takes the energy from ATP, grabs one of the phosphate groups, and it attaches that to glucose. Now, when you attach it to glucose, to the 6th carbon, it becomes glucose 6-phosphate. And then this molecule is unable to leave through the same transporter because now it has a completely different shape and identity. Therefore, glucose 6-phosphate becomes trapped inside the cell because there is no transporter for glucose 6-phosphate. The next step of prominence that I wish to bring you is step 7. And during step 7, we have a manipulation where 2 phosphate groups are present on the glycerate, but after the step, one of the phosphate groups has been removed and given to ADP. So ADP becomes ATP, and this is accomplished by this enzyme, phosphoglycerate kinase. And a kinase is any enzyme that moves phosphate groups from one entity to another. Now, the other thing that students need to understand is that there's a special name for this type of manipulation of ATP. When ATP is made, not by another system that we'll discuss later, but directly as a transfer from one molecule to another, we call that substrate level phosphorylation. Substrate level phosphorylation. And as I said earlier, that will become clearer once we encounter the second type of phosphorylation inside cells. So combining the knowledge from step 6, which was mentioned earlier, and the last slide, which is step 7, we find that these 2 steps are very important when it comes to moving energy around. So remember, energy is stored in the bonds and also in the electrons within the glucose molecule. So step 6 is responsible for capturing some of the electrons and then transferring them to the energy carrier NAD, making that NADH. And step 7 is responsible for capturing energy stored in the phosphate groups and then passing that on to the ATP carrier molecule. Why are steps 6 and 7 important during glycolysis? Well, if you view the energy levels between molecules and the energy carriers using a graphic such as this, the brown arrows going down are to do with the energy released by the glucose intermediates in the glycolytic cycle. So here we have the substrate, and there we have the product. And then here we would have the substrate, and there we'll have the product. But let's concentrate on step 6. So step 6 releases enough energy that it can do two things. It can form NADH from NAD, i.e. transfer electrons and protons to NADH. But there's still some energy left over to form this high energy bond between the phosphate group and the aldehyde. Likewise, in step 7, the energy released by breaking this bond is enough to charge ADP with ATP. So these are energetically favorable reactions. And as we know from chapter 3, this process of changing one molecule and at the same time changing another is called coupling. So here we have two couplings, here we have one coupling. And of course there's also some heat generated which takes care of the rest of these arrows. So this is step 10 of glycolysis, where pyruvate is generated, and this is pyruvate here. And you can see the substrate here for this enzyme, pyruvate kinase, has a phosphate group, but pyruvate does not. And based on what we just learned in the previous slides, we can imagine what happens. The removal of this phosphate group releases a lot of energy, enough to charge ADP with a phosphate. In most cells, the process of glycolysis does produce pyruvate, but the question is, what happens next? In some cells, there's no choice. They cannot live in the presence of oxygen, and therefore they have to enter the left pathway here. But for other cells, there is a choice. In the presence of oxygen, they enter the right pathway, and if oxygen is limiting or absent, then only then will they enter the left pathway. So the left pathway is fermentation. And as far as we're concerned, there's two types of fermentation available to living systems. Lactic acid production, which is in our cells, our muscle cells. They can ferment when oxygen is missing in order to keep the muscle running and to take you away from danger. But in other organisms, the fermentation process is missing these enzymes, but it has evolved alternative enzymes. And in that case, alcohol, ethanol, and carbon dioxide are the byproducts of fermentation. And yeast cells use that, and humans make use of that for making alcohol and for using the carbon dioxide for making bread. Summarized in this slide, we have those two processes. So your muscles can do the above in panel A, and certain other organisms can perform panel B in the absence of oxygen in both cases. Interestingly, you can see something going around in the cycle here. So NAD plus, which is the depleted energy carrier, is then converted to the charged energy carrier in glycolysis. But during the conversion from pyruvic acid to lactic acid, the electrons are taken from NADH and given to lactic acid. So NAD can then recycle itself. If this step is prevented, then all the NAD in the cell will be NADH, and the cycle will come to a standstill. Likewise, if you study this in more detail, you'll see that the same process is taking place down here, except that the alcohol here only has 2 carbons, and the third carbon is lost as carbon dioxide, whereas lactic acid has 3 carbons from the pyruvate. So studying these fermentation reactions more critically at the level of the NAD, we find that during glycolysis, energy electrons are given to NAD plus. So NADH is carrying energy, much more energy than NAD. So when pyruvate is converted to lactic acid, the lactic acid will have more energy than the pyruvate. Why? Because the high energy electrons carried by NADH are given to the pyruvate, making it more energetically charged. The reason we're doing that is because we need to recover our NAD plus so it can continue to keep glycolysis running. Should this step become poisoned, then NADH would become accumulated in the cell and NAD plus depleted, and this step of glycolysis would come to a standstill, and in that case the muscles will completely stop. The same thing happens in the production of alcohol and carbon dioxide in yeast cells. The rationale is the same. So ethanol contains more energy than the pyruvate from which it's made through a two-step process. Let's return to our understanding of what happens in cells that do have oxygen available to them. In that case, the reactions of glycolysis are continued to be pushed into the mitochondria, where the citric acid cycle enzymes reside. So the pyruvate is pushed into the matrix of the mitochondria. So students should be familiar with the anatomy of a mitochondrion, as indicated here. So here's a quick summary of where we are. So glycolysis was taking place in the cytosol of the cell, and through a 10-step process, pyruvic acid or pyruvate was manufactured. The pyruvate is now pushed through the double membrane of the mitochondria into the mitochondrial matrix, where it will then undergo some additional changes and then be fed into the Krebs cycle or the citric acid cycle. The pyruvate is transported through a transport protein located on the inner membrane of the mitochondria and converted to acetyl-CoA. Now, acetyl-CoA has 2 carbons, not 3. So what's happened to the third carbon? Well, this is not a carbon here. This is just acetyl-CoA. So the third carbon is actually lost. It's lost as carbon dioxide in step number 1, which we'll see shortly. So step number 1 cuts off one of the carbons and the 2 oxygens right here to make carbon dioxide. And then that carbon dioxide diffuses back into the cytosol and then back out of the cell into the bloodstream. Now, in this process, energy is released, more energy is released, and that's picked up by another molecule of NAD or NAD+, becoming NADH, a charged electron carrier. And coenzyme A is brought in and attached to the remaining part of the pyruvate to form acetyl coenzyme A. The steps we just covered of production of carbon dioxide, the charging of NAD+, and the joining of coenzyme A are all performed by a giant complex of enzymes. So that's called the pyruvate dehydrogenase complex. It's actually 3 enzymes all in one lump. And each part of that mega enzyme does its own job and then passes the products to the next. So in reality, this is a better depiction of what it looks like. So there's 8 trimers of this enzyme, there's 6+ dimers of this enzyme, and there's 12+ dimers of another enzyme. So this is called a machine, and once the machine does its job, the end product is acetyl coenzyme A. And what acetyl coenzyme A does, it then feeds into the TCA cycle or the citric acid cycle. So in reality, where are we now? So we said to you earlier that sugars are digested and then absorbed into the body, then transported to the cells, where the sugars are taken in across the membrane through transporters, converted into glucose 6-phosphate, then undergo another 9 steps to become pyruvate. The pyruvate is then transported in the presence of oxygen into the mitochondria matrix, where the pyruvate is converted through a sequence of steps, as we just learned in the previous slide, by the dehydrogenase into the acetyl-CoA. Now what this slide is saying, that other food categories, other food classes like fats and proteins can also be fed into various steps in the pathway. And we'll see that coming up shortly. But for now, what happens to fats? Well, fats can be stored inside the cytosol as droplets, and that's indicated here. So here's the cytosol. Here's another, this is an organelle, and here we have a drop of fat. Now, what does this fat molecule look like? Well, it's actually something you're familiar with. It looks like a phospholipid, but not quite. We have the glycerol part here. Glycerol is 3 carbons. And then we have fatty acids attached to the glycerol through ester bonding. So this is not a phospholipid, because a phospholipid would have 2 carbon tails, and then the 3rd carbon would be attached to a phosphate. In this case, all 3 carbons are attached to fatty acids, and that gives us our triacyl glyceride, the most common way in which fats are stored as droplets inside cells. So the fats are the second most favorite molecule of energy after carbohydrates. So fats are likely to enter the citric acid cycle at certain points, depending on the fat, and they are then chopped up, their bonds are broken systematically, and as the bonds are broken, the energy is transferred to NAD plus to form NADH, and another carrier molecule called FAD to form FADH2. And these electron carriers then take the energy away to somewhere else. In the process, every time the cycle spins, carbon dioxide is generated, and then that carbon dioxide is released into the cell and then into the bloodstream, just like we learned earlier. So let's have a look at the citric acid cycle as a review. So what goes in and what comes out. And it's a cycle, so it spins round and round, and stuff comes out at various locations. So what goes in? As we learned earlier, acetyl-CoA, the 2-carbon molecule, enters the citric acid cycle, or the Krebs cycle. And what comes out? Well, those carbon atoms are split from each other, forming carbon dioxide. Also, ADP is charged to ATP. So a phosphate group must be grabbed from somewhere here and given to ADP. Also, 3 NAD pluses are charged with electrons and protons, as well as 1 FAD charged with protons and electrons. So a lot of energy is pulled out of the Krebs cycle, and that energy comes from the bond holding the 2 carbons and other atoms in place. Here is the Krebs cycle in more detail, the citric acid cycle, or the TCA cycle. So step 1 is where the acetyl-CoA, the remaining PartA pyruvate, enters the cycle. And then that is systematically changed into various substances, and parts are pulled apart in 8 steps. So the first thing to be produced is citrate, and then that becomes isocitrate, etc., etc. So the students will need to learn the parts of the citric acid cycle with the names of the enzymes, and of course the names of the products and the substrates, and also where energy is pulled out. So step 3 results in 2 products: a charging of an electron carrier, NADH, and the removal of one of the carbons that came in with the acetyl-CoA. Step 4 charges another NADH, and it removes the last carbon that we had earlier. So all the carbons are now removed from food molecules and eliminated as carbon dioxide. But still, the molecule has some energy invested in it, and that energy is used to make ATP via GTP. So maybe this is the first time in this course that we have come across GTP, but just keep in mind that the GTP will then be handing its energy over to another enzyme, which makes ATP. And then at step 6, we have FAD being charged with energy. But still, there's energy left, and once we manipulate the molecule one more time, then that energy can be pulled out in step 8 to produce another NADH. So 1, 2, 3 NADHs, 1 FADH2, 1 ATP, are produced as energy molecules, and then 2 waste products, carbon dioxide, are released. And the cycle then repeats again and again and again, as long as it's being fed from 12 o'clock by more acetyl-CoA. So here we have a nice chart relaying what's going on. So remember that glucose had 6 carbons. Where have all those carbons gone? Well, we have to prove that all 6 carbons end up in carbon dioxide. So let's have a look. So during glycolysis, none of the carbons are removed. They are rearranged, but none are removed. During the conversion of pyruvate to acetyl-CoA, 1 carbon is removed from each molecule. But remember, 2 molecules of pyruvate are produced from each glucose, so we have to multiply this by 2. So 2 of the carbons have been removed, leaving 4 from each glucose molecule. And during the citric acid cycle, the remaining 4 molecules, 2 each for each of the acetyl-CoAs, are removed. So all 6 carbons that were present in glucose end up as becoming carbon dioxide. So how many of these energy carriers are charged? Well, there's 2 types here that we talk about, right? NAD and FAD. So NAD, we get 10 of those produced. 2 in glycolysis, 2 in the conversion to acetyl-CoA, and then 6 during the TCA cycle, or the Krebs cycle, citric acid cycle. And finally, we only get 2 FADs being produced in the TCA cycle, and none during the previous steps. So only 2 of those are produced. So 6 waste products, 10 high energy NAD molecules, and 2 high energy FAD molecules. On this slide, we offer a slightly more detailed molecular view of what's happening to the substrates as they are being manipulated around the cycle. So students should look for the logic in what's going on. And if you look very carefully, nature is very good at doing things in stepwise fashions. Not too much in each step, just enough to manipulate the molecule by its enzyme. Now, the Krebs cycle is named after Hans Krebs, who in the 1930s performed a lot of experiments on tissue from mice, and he was able to elucidate what was going on. So he is given credit for the Krebs cycle. Other than producing energy, as we'll see shortly, the Krebs cycle and glycolysis combined are responsible for producing branches in a complex biochemical dance. So the pink line that you see here is the glycolytic cycle and the TCA cycle. And once we study that in detail, we find that from various parts of that, molecules can be sequestered to make almost everything the cell needs, from cholesterol to nucleotides to amino acids to chlorophyll if this was a plant cell, to heme groups in blood cells, etc., etc. So that's why we have the importance dedicated to understanding and learning glycolysis and the citric acid cycle, because of their key role, their central key role in living cell biochemistry.